Guten Abend, liebe Gäste. Ich darf Sie herzlich begrüßen zur Vernissage, zu der uns heute Ayelén Valko eingeladen hat, mit dem Titel Zelluläre Welten. Eine Reise mit dem Blick durch das Mikroskop und für mich durch die Brille hat sie uns eingeladen, um uns darzustellen, was ihre eigentliche Arbeit ist, nämlich die wissenschaftliche Kommunikation. Ayelén wird nachher erklären, was wir hier sehen. Ayelén ist Wissenschaftlerin. Sie ist promovierte Biologin, hat sich spezialisiert auf Zellbiologie, aber sie hat auch wissenschaftliche Illustration in Buenos Aires studiert. Mit ihrem wissenschaftlichen Hintergrund lässt sie uns teilhaben an einem Prozess, der Unsichtbares, eigentlich Unsichtbares sichtbar macht. Sie beherrscht eine Reihe von Techniken, darauf werde ich nachher noch eingehen, auf ihre Materialien. Was wir hier sehen, das fällt uns gleich auf, ist keine freie Kunst, wie wir sie normalerweise sehen. Da ist kein schneller Duktus, da ist viel Präzision, eben die Präzision einer Wissenschaftlerin, in der sie denkt und arbeitet. Zum Zweck der wissenschaftlichen Kommunikation erstellt sie wissenschaftliche Illustrationen, die komplexe Inhalte verständlich machen. So können Konzepte, Strukturen und Prozesse, Aufnahmen aus Licht- und Elektronenmikroskopie auf eine Weise vermittelt werden, für die Worte alleine nicht reichen. Dies gilt für Fachleute wie für Laien gleichermaßen. Es darf sich eine Schulklasse des Helmholtz-Gymnasiums freuen. Sie werden unterwiesen in diesem Zusammenhang aus Kunst und Wissenschaft. Und Ayelén hat auch sehr viel Erfolg, viel Anerkennung mit ihrer Arbeit. So hat sie mit Freuden den Zuschlag bekommen, ihre Arbeiten auf wissenschaftlichen Journalen vorne auf der U1 auf dem Cover zeigen zu dürfen. Die sehen Sie hier hinten in den Rahmen und die sehen Sie aber auch hier im Original. Ja? Da können Sie gerne reingucken, drin blättern, aber wir würden Sie gerne bitten, diese nicht mitzunehmen. Das sind Ihre einzigen Exemplare und da wäre sie bestimmt traurig, wenn die wegkämen. Diese Cover, die sie hier so schön präsentiert und gerahmt hat, die kann man auch kaufen für übrigens, finde ich, sehr kleines Geld. Also nicht nur die Kunstwerke, sondern auch diese Darstellungen. Jetzt, was ist eigentlich die Kunst an sich? Also natürlich das Handwerk, das darin liegt, aber es ist auch die Art, wie sie Dinge kombiniert. Sie bringt Dinge in einen Kontext, in eine Ebene, die so nicht in einer Ebene sind, um einfach Inhalte zu verdeutlichen, in der Absicht, sie besser zu verstehen. Und dabei nimmt sie sich auch die Freiheit, mal psychologische Momente wie Traumata einzuarbeiten, die man eben in ihrem sich auflösen, das weiß man heute, in ihrem Auflösungsprozess, die Traumata visuell erkennen kann. Und sie wird nachher zeigen, I'm talking about psychology and the trauma, so how it can the whole problem can be resolved after a while, and yeah, you will mention that, talk about it. Ausgehend vom ganzen Organismus, vom gesamten Organismus, begleitet sie uns oder lässt sie uns teilhaben auf einer Reise in das Zellinnere, in die Zellorganellen, in den Zellkern bis hin zur DNA. Da werde ich noch drauf eingehen.
Well, thank you, Angelika, for that kind introduction of my work. And well, again, thank you all of you for being here with this rain. So, can you hear me? Yes. Okay, well, so as you can see, this is an exhibition inspired by science, right? This is an art and science exhibition. So, something that I think that is very, not very, but quite common is basically to find, for example, cover images, as you can see there, right, as those that I created there or here, because it's usual that science uses as a tool art basically to communicate science, right, as a science communication tool. But what is not so common is that basically art finds inspiration in science basically for artistic creation, right? That is basically not so common, and that is what this exhibition is about, basically. So the way in which basically we thought this exhibition was as a visual journey from entire organisms, as Angelika was saying, toward the interior of a single cell, right? So here in all the exhibition you will find like different organisms across scales, right? So I mean entire organisms, organs, tissues, cells, and also basically cellular landscapes, right? What is happening inside the cell, and even molecules. So I'm basically, well, Angelika already said that, but I'm a cell biologist and also an artist, right? So that is basically that is the reason why I'm basically depicting all these things, because during my PhD I was trained as a microscopist, right? I was trained on different microscopy techniques. I know fluorescent microscopy, electron microscopy, different microscopy techniques, which allowed me to access to the fascinating subcellular world, right? That is, basically those amazing images are in general restricted to the scientific community, right? Not to the general public. So I thought it would be a good idea basically to depict those also for the general public, right? To share that beauty and mystery hidden inside a cell. So basically I painted and I'm drawing and painting all my life, right? But during the PhD, I realized that that was a very good idea, actually, to get inspiration, artistic inspiration from cellular landscapes, right? So as Angelika said, basically, I depicted here some like different model organisms, different organisms. I worked during my PhD using the fruit fly, Drosophila melanogaster. So you will find, I know that it's a little bit crowded, so when you walk around, you will see that there are different stages of Drosophila melanogaster life cycle, the fruit fly life cycle there, and also there. And you will find as well different organs, right? For example, in that wall, you will see afterwards, I mean, it's a little bit crowded, but I try to represent also organs inside the fly, right? The hematopoietic organ of the fly, for example, that is that black painting that you can see there, is basically the organ responsible for basically producing blood cells, and also, for example, the salivary gland of Drosophila. And yes, there you see, for example, a larva, a fruit fly larva, in which I highlighted an organ inside, right? Because the idea of all the exhibition is going inside, going deeper and deeper inside organisms, right? So a process that I studied during my PhD, I would say that we use model organisms to study different like biological processes, right, in science. So during my PhD, I used this fly, Drosophila melanogaster, to study a very curious process called autophagy. I don't know if you have heard about that process. I know that some of you did. But yeah, basically it's a very interesting process, philosophically speaking, because it's a self-eating process. It's a destructive process that happens inside the cell that yet is necessary for survival. So when cells are exposed to stress stimuli, they basically eat themselves, right? Like stress stimuli are, for example, fasting, right? So they eat themselves, and they need, because they need to obtain nutrients and energy from somewhere, right? So they eat themselves. But that process, that sounds horrible, is necessary for survival. So I think that it's a very interesting process for artistic creation, right? That apparent paradox, right? Between destruction and creation somehow. So that wall that is there basically represents that process, the autophagy process, across scales. So in the back, in the bottom, sorry, you can see an organ in which I basically highlighted the autophagy process, adding some like plastic pearls, some plastic beads, right? Because during autophagy, cells form new compartments inside the cells that are vesicles, called autophagic vesicles, where different cellular components are trapped and then degraded there, destroyed there. So the plastic bits actually represent each single vesicle, right, that is formed inside the cell. Actually, that tissue is, basically the painting represents a tissue called a fat body that is basically the liver of the cell. So that is a tiny part of the liver, the liver of the fly, sorry, a tiny part of the liver of the fly, right? It's very tiny. So, and the other paintings that you can see in the wall are actually zoom-ins of the previous painting. So you can see the autophagic vesicle closer and closer, right? So in this painting, for example, that we are seeing here, I'm representing something different, right? That is the fact that cells are composed of different organs as us, right? Called organelles. So there are different compartments that have different functions in the, on the cell. And this is my representation basically of an yeast cell. I work with yeast cells during my postdoc. And basically I try to represent the ER, the endoplasmic reticulum, that is an organelle responsible for producing, creating proteins in the cell. It's a very important organ. And I try to represent there how this organelle is created and destroyed, right? That is the reason why the painting is called equilibrium. So there is an equilibrium in these two sides of the cell between creation, where you can see a lot of ER in green and red, and destruction on the other side, where you can see that there are like a few tubules, right? So this is basically a very interesting model organisms, and actually I try to represent artistically what was happening inside the ER in the painting, the big painting, the large painting that you can see there, the black one, that is basically representing a very, very, very small fraction of the membrane of the ER. So it's basically what is happening just in the membrane. So it's basically a tiny dot, right? So I have reached there the level of molecules, basically, inside the membrane. And basically to finish this like brief description of the exhibition, I would like to focus on these two series. These series are not finished yet. I'm still working on them. But the reason why we placed this series here is because, these two series, is because they talk about somehow opposite processes, right? This series here talks about catabolism, about biological destruction. And that series there talks about anabolism, about biological creation. So here, I introduce, obviously, there aren't humans inside the cell, right, of course, but I try to represent here basically the stomach of the cell that is called lysosome. It's an organelle as well, a compartment. And basically I used different techniques here to highlight different features of this organelle, right? And I placed some humans inside as metaphors, right, as a symbolism, basically to talk about like the destruction that takes place in this organ that is like a stomach, right? So this series is not finished, but what I'm planning to do with this series, this is like a spoiler alert, it's basically I'm going to degrade the humans inside completely. So the last painting will be just the lysosome without humans anymore, because this series talks about destruction, right? But this series, because not everything is negative, right? This series talks about biological creation and represent basically an ovary, right, across scales. So basically, these are like zoom-ins, right? So the journey begins here with the ovarian tissue, and then it's going there. So in the ovarian tissues, you have different like egg cells, as you know, like follicles. So then I'm doing like a zoom-in of this follicle, of this egg cell, that is this one. Then I'm going inside the cell, here. So here you can see this is the nucleus, and this is the nucleus, right? And at basically the last painting is this one, that is what is happening, according to my artistic interpretation, inside the nucleus. And what is interesting, as Angelika was saying, is that in these two series in which I'm using metaphors, I'm trying to blend, I'm trying to mix psychology and biology here, right? Because this talks about like internal processes, right? About, I don't know, the relationship with others, about traumas, about fears, about a different way to deal with reality, right? And I'm mixing those ideas with biology as well. So,